So today I'm going to talk about the labs that we will be doing this semester. And to talk about this, I'm going to start at really what was close to the end. This is a picture from last year where a group of students and I traveled to Western Kentucky University to do electron microscopy to see pictures of the phage that they had isolated over the course of the semester. And so here you can see on the computer screen the phage that one of these groups of students, and really I don't remember who, had isolated during the course of their time over the semester. But that's really close to the end. So we need to talk about what it is you're going to be doing this semester. This is a nationwide project that is sponsored by the Howard Hughes Medical Institute, which is a really large research organization that is also heavily focused on undergraduate education and improving undergraduate biology education in particular. And so as part of this project, which is called SEA PHAGES, what you're going to do this semester is to isolate phage from the soil. So phage or bacteriophage, those two terms are used interchangeably, are viruses that infect bacteria. So in brief, what you're going to do this semester is isolate and purify phage from the environment, and then do a little bit of characterization of these phage. Next semester, in the spring, there will be a second course where students will analyze the genomes, the DNA of these phage, to figure out what they do. And that is an elective course that you are all invited to enroll in, and we'll not talk about that now because next spring semester is a long way from now. So here is a flow chart. This flow chart is in your phage discovery guide, which is the lab manual for this semester, and that's available on Blackboard. So what you're going to do is you're going to start by isolating the phage by collecting environmental samples, which sounds really fancy, but really what this means is you're going to go out and you're going to dig up a sample of soil. I should say that we're streamlining the process a little bit this semester because of the conditions we are working under. So you will carry out an enriched isolation from these soil samples to look for phage. So you'll do some tests to determine if you have phage, and when you do have phage, then you move on to a purification stage to make sure that you have a population where all the phage in that population are identical to each other. When you have done that, we move on to an amplification phase, where essentially you just make lots and lots and lots of phage so that it can be characterized. And the phage are going to be characterized in three main ways. You're going to take the phage and you're going to extract DNA from that phage and look at that DNA. There's two ways to look at that DNA. One is by cutting the DNA with enzymes and running them on a gel so we can look at them. Some of you probably have run these types of gels in your high school classes. And another way to characterize the phage is actually take that DNA and sequence the DNA. So the DNA sequencing will be happening over winter break, and the sequences will come back and they will be analyzed in the spring semester class. The other piece of characterization is microscopy, and that's the picture that I showed you at the beginning. I'm not sure at this point how the microscopy is going to go. I sincerely doubt, unfortunately, that we'll be able to take a trip to Western Kentucky for you all to do electron microscopy by yourselves, but hopefully we'll still be able to get photographs of your phage so that you can look at them and look to see what these phage look like. So for the remainder of this lecture, we're just going to talk a little bit about phage and give you a little bit of basics on phage biology. This video is supported by the material in Chapter 3 of the Phage Discovery Guide. So this is a picture of a phage. To me, these phage always look a little bit like alien spaceships, but this really is what they look like. So phage is composed of a head, as we see here. Inside this head is the DNA or the genome of the phage, and then you have this tail. And what the tail is going to be used for is for the phage to attach to its host cell. And because these phage infect bacteria, the host cell are bacteria. So remember, these are viruses. So different viruses have different types of shapes, and this is kind of the general shape of a bacteriophage, but you probably have seen in the media pictures of the coronavirus, which is also a virus, and it has some similarities to this structure. So how does a phage reproduce? We see that here in this life cycle of a phage. There are actually two different types of life cycles, and we'll talk about this one first because it's simpler. So what happens is the phage attaches to a bacterial cell, as we see here in stage 1, and it injects its DNA into the host cell. We get here into stage 2. That phage DNA circularizes, and then the genes encoded on the phage genome get expressed. And what happens here is that the host cell DNA that we see here in black gets destroyed, and the phage DNA that we see in blue gets replicated. So you get lots and lots of phage DNA, as we see here in step 3. And also, the host cell is used to make the rest of the components of the phage, so the protein components, which are the head and the tail. And so we see here that we have phage heads and phage tails and the DNA, and this all gets packaged together. And so here we have the packaging. So now you have multiple phage particles found inside this bacterial cell. And in the last step, the phage causes the bacterial cell to lyse, in other words, to burst open, and these phage are then released where they can go on to infect new bacterial hosts. And so in practice, what this means is that if you take bacteria and you grow it on a plate, what happens is the bacteria grows on a plate, and you see a bacterial plate that is cloudy with bacteria on it. So this plate here has all this haziness, and that's where all the bacteria has grown. But you see these clear spots on the plate, and these clear spots are where a phage has infected a bacteria and then caused that bacteria to lyse, releasing the phage to the bacteria surrounding, and then those surrounding bacteria also lyse, and so on. And so what you see then is a clearing in this otherwise hazy lawn of bacteria. So these clearings are called plaques, and what the plaques are an indication of the presence of phage. Right? So there are millions of phage in each plaque. So this is what we call the lytic life cycle of the phage, where the phage infects the bacteria and causes it to burst right away. There is an alternative life cycle that some phage can undergo, and that's called the lysogenic cycle. And it is temperate phage, phage that are called temperate phage, that can do the lysogenic cycle. So temperate phage, when they infect a bacteria, as we see here, what happens is they come to a fork in the road, and they can decide what to do. One of the things they can do is to lyse the bacteria, as we've talked about. But the other thing that can happen is that the phage genome gets integrated into the bacterial genome. And so you can see that here, the phage genome in pink and the bacterial genome in gray, and you can see that that pink genome has been integrated. In this lysogenic state, the bacterial cell does not lyse, and when this bacterial cell divides, the DNA is copied, and all the DNA is copied, including the DNA in that phage. And the phage can stay in this state for quite a while. But at some point in time, it can switch back to the lytic stage, as we see here in this life cycle. So here it's in that lysogenic stage, and it can be maintained here for a long time. But what can then happen later is that this arrow gets reversed. The phage DNA, the phage genome, comes back out of the bacterial genome, and now the phage can undergo the lytic cycle, as we've talked about. For these temperate phage that can undergo lysogenic life cycle, their plaques can look a little bit different, because what you have, instead of all the bacteria that have been infected by the phage lysing and forming a clear spot, what can happen instead is some of that bacteria can grow in this lysogenic phase. And so what you might see, instead of these clear plaques that we looked at earlier, you might see cloudy plaques, as we see here. I should note here, though, and that's pointed out here in this figure legend, that there are examples of phage that produce cloudy plaques, but still under our experimental conditions don't appear to form lysogens, that is, don't appear to integrate their genomes into the bacterial cells. And really the bottom line is we don't understand very much about phage at this point. And that brings us to you and the whole point of this semester's experiments. There are lots of phage out there. Most of them have not been characterized. And there is a vast amount of really interesting biology that is waiting for us to discover. And what we're doing here in this semester is allowing you to be part of this discovery process. So over the course of the several years that we've been working on this phage project here at UE, we have isolated a whole bunch of phage, and I'm just going to show you two of them. This is CRAF. CRAF is a phage of the Syphoviridae class, and the sypho here means siphon, and so you see here that it has a really long siphon-like tail. And so here are these pretty pictures of CRAF that my student Skip took when he went and did electron microscopy. And Skip has now graduated from UE. He just graduated last spring. He's now pursuing a PhD in biology, and hopefully I'm remembering this correctly. I think he is at the University of Colorado. Here's a very, very different looking phage. This is of the class called Myoviridae, where you see the tail is very, very much shorter. And it's hard to see the scale in here, but we have a really big phage head. And so Ian Alberts, when he saw this phage that he isolated, he looked at it and he's like, wow, it's got a really big head. Let's call it Megamind. So that's the other thing I forgot to say, which is that when you find your own phage, you get to name it. And so Ian named this Megamind. Ian is a senior here in the biology department and will be doing some tutoring for Biology 119, and I'll talk more about tutoring later on. So to finish up, let's talk about some fun phage facts. There are an estimated 10 to the 31st number of phage on Earth. If you took all their genomes and strung them out together, you'd get a distance that is 42 million light years. So that's enormous. It's a number that to me is pretty much unfathomable. That so many phage, 10 to the 31st, but really only about 3,000 of these phage genomes have been sequenced. And so you can see that there is so much information out there that we can discover because there is so little that has been discovered about phage. Most of these 3,000 genomes actually have been sequenced under the context of the SEA PHAGES program that we are part of. So these phage are constantly doing the thing they do, which is infecting bacteria. There are 10 to the 24th infections of bacteria per second. And so this might raise this question in your mind, if all these phage are out there infecting the bacteria, how are there still bacteria? And so here is where we talk about this kind of arms race between the phage and the bacteria, where the phage are infecting the bacteria and causing them to lyse, and the bacteria are trying to protect themselves from the phage. And so some really important biotechnology tools like restriction enzymes and CRISPR systems evolved in bacteria to cut the phage DNA to fight against phage infection. So restriction enzymes are enzymes that cut DNA at specific places. And we use them a lot in biotechnology. You might have used them in your high school classes. And we will use these restriction enzymes towards the end of the semester to analyze our phage DNA. CRISPR systems you might have heard of, or you might have heard of genome editing, which has really become important in the news in the past few years. And this is a system that hopefully we'll have some time to discuss in greater detail later in the semester. These are systems that can be used to very precisely change DNA sequence. And so there's a lot of excitement about being able to edit your genome and perhaps edit out a mutation that is causing a disease. And whether these come from, well, they came from bacteria. Why do bacteria have them? Essentially to protect the bacteria against phage. So we looked at Megamind and I said that it has a really big head. The phage head is also called a capsid. So when you see a really big capsid or phage head, what you can predict is that there is a very large genome inside that phage. And that is what we did see when we eventually sequenced Megamind. So all that DNA gets packaged into that capsid, and it's packaged at really high density. It's the same pressure as a bottle of champagne. And so I guess you could imagine like popping that capsid open and all that DNA bursting out. The majority of the DNA in the phage genome, and this is something you'll discover if you take next semester's phage genomics class and try to characterize that DNA, what you find is that most of that DNA codes for unknown proteins. And so there's, like I keep saying, lots out there that is unknown that could be really useful. I'll end this slide by talking about much more recent discoveries. I want to point out that what you're going to be doing this semester is isolating phage that infect bacteria that are found in the soil. And so we will be working with the soil bacteria in the lab, and that soil bacteria is non-pathogenic. It doesn't cause human disease as far as we can tell. There has been no evidence that this causes human disease. But of course, you know that there are lots of bacteria that do cause human disease. And right now, what we have in our medical arsenal to fight bacteria that cause disease are generally antibiotics. And you've probably read about the problems of increasing antibiotic resistance of bacteria. What happens is these bacteria, because they are exposed to all these antibiotics, have evolved defenses against the antibiotics, and so the antibiotics don't destroy them anymore. What other types of defenses do we have against bacteria? The short answer is not many. But phage could be a defense against bacteria because, as we have just talked about, phage can burst these bacteria and break them open so that they die. So this was exciting news from last summer, so May of 2019, where we have this news report that shows that this teenager here was treated for a bacterial infection using phages. And so these phages were actually isolated as part of the SEA PHAGES project that you are about to begin. And so what happens at the end is you get all these phages, and we send all the phages to be archived at a freezer at the University of Pittsburgh, where the person in charge of this project is a professor. And so what they did actually was they went and looked at the phage collection in the freezer and tried to find phages that could lyse the bacteria that was giving her this bacterial infection that was antibiotic resistant. And so you can see here from the headline that this phage therapy was successful. And so we were really excited about this, and this is a really cool application of the phage work that you are doing. So the big question that we're going to discover this semester is, what are your phage going to be like? So to end this lecture, I just want to give you a quick snapshot of what you're going to do next. So there are two options for phage isolation. One is the direct isolation, where you try to find all the phages in the sample. It does have a lower probability of success because your soil sample may have very few phage in it. And in a direct isolation, you might not be able to capture those phage. The other option is an enriched isolation, where what you do is you take the phage from the sample and then you try to amplify them before testing. This gives you a higher probability of success, but it's not necessarily a representative population of phage that you're going to get, because some phage in the sample might grow better than others, and the ones that grow better are going to be favored. Because of the constraints we're working under this semester, we are only going to be doing enriched isolations. And so your next step for about a week from now is for you to collect a soil sample. So just go out into campus or home or wherever you are and collect a nice soil sample. We will give you collection tubes for that soil sample. And when you collect the soil sample, you should record the characteristics of that sample and the GPS coordinates of the position where you collected the sample. And you're just gonna put the soil in the tube and cap that tube so the soil samples don't dry out, and drop your sample off to me or to Dr. Merkel before noon on Thursday, September 3rd, because you will be doing the enriched isolation on Friday, September 4th. So that's it for this introduction to the phage project. I'm really excited to be working with you this semester on this, and I can't wait to see what we discover.